Sally Hind. I'm from UK HSA, and this poster is introducing a platform technology to counteract efflux in antimicrobial scaffolds. So this is a collaboration between UK HSA, the microbiologists there, and the medicinal chemists led by Miraz Rahman at King's College London. And it's a reimagining of the classic EPI strategy, where you have one compound which interacts with the efflux pump, and you have your novel scaffold which is subject to efflux, and they compete at the efflux pump, and therefore you get more accumulation of your compound in the bacterial cell. But this hasn't translated to the clinic because you need such high concentrations of the efflux pump inhibitor that you get off-target effects and toxicity issues. This technology, we use computational modeling to identify small pharmacophores which interact with the hydrophobic pocket of the efflux pump. And then we attach those synthetically to the chemical scaffold that you know is being effluxed. So it's all one molecule. So therefore, when this molecule interacts with the efflux pump, the little pharmacophore interacts with the hydrophobic pocket, the conformational change doesn't happen in the efflux pump, and the whole compound bounces back into the cell, and you get higher accumulation of your drug inside the cell. So we have applied this initially to fluoroquinolones, which we all know are effluxed heavily out of both gram-positive and gram-negative bacteria. We've also successfully applied it to the azole class of antifungals, and therefore resensitized Candida auris to fluconazole. And we also have a novel scaffold, which is our gram-negative lead, which is in development at the moment and is part of an MRC grub. So this poster covers the proof of concept for the fluoroquinolones. So we initially targeted gram-positives as an easy start. We used levofloxacin as the scaffold and Norray as the efflux pump to demonstrate this technology would work. We assembled a panel of Staph aureus strains. The first two strains are sensitive strains, so they've got an MIC of 0.125 to levofloxacin. Now, two lead ERB compounds have really low MICs, like 0.08 and lower. This strain here is a isogenic relation to the strain above, but it just has upregulated Norray, no other difference. And you can see that the MIC to levofloxacin increases to reflect this increased efflux of the scaffold out of the cell. But for our two ERB versions of levofloxacin, there isn't any jump in MIC, suggesting that they are retained within the cell. They're not effluxed out through that upregulated Norray. The last two strains have that upregulated Norray, but they also have the mutated target, so the gyrase, and the levofloxacin becomes useless in these strains, whereas our compounds, although there is a slight increase in MIC, they're still at clinical levels, so that's really positive. We used a gyrase biochemical assay, so this is cell-free, to demonstrate that the two ERB compounds have a very similar IC50 against this target compared to levofloxacin. But when you look at the fluoroquinolone resistant version of gyrase, levofloxacin IC50 jumps from 8 to 200 micrograms per mil, whereas the IC50 for our two lead compounds, especially SBL7, there's barely any change. This suggests that the compounds with this additional little pharmacophore are binding slightly differently to the target. And when we've done resistance evolution studies, we've seen mutations in a different part of the gyrase, so they're kind of confirming that result. We used Galerium melanella to look at efficacy of the lead compound against a MRSA strain, and saw good efficacy, very similar to levofloxacin, and this translated to a mouse model. We've also got really strong safety data for this, so there's no hERG inhibition. The C450 assay is clean. So we've now, as I said earlier, we've also applied this to the azoles, and I've got one demonstration of an MIC table here for a Candida auris strain, which has mutations in the target for the azoles, and it has a selection of efflux pumps which actively efflux out fluconazole. And the fluconazole MIC in this strain is 1 to 8, so it's completely useless. Whereas our lead compounds, which we've designed again specifically for the efflux pumps in Candida auris, have really low MICs. And we've got very similar selection of data, and that's gone through animal studies as well, and we've got the safety profiles. And the one that we're now concentrating on is our gram-negative lead, which is a novel scaffold, and we're very excited about that. Thank you to the internal funding from UK HSA through the Open Innovation Grant for supporting the development of this technology, the MRC for funding our new gram-negative lead, and BBSRC for a PhD project as well. So thank you.